Hi guys, I'm Murlidharan Nair. I'm an associate professor of bioinformatics at Indiana University, the regional campus. And this is a joint work with my collaborator, Professor Jenny Prosperi, who is at the IU Medical Center and at Notre Dame as well. And the title of my talk is Genome-wide transcriptomic analysis of APC knockdown and exposure to chemotherapeutic drug in breast cancer cell line. So let me briefly go through a little bit about what cancer is. This is just a figure that I've taken from Hannan and Weinberg, the classic paper that everybody uses as their first figure when you're talking about cancer. So essentially, cancer is a disease of the genes, and it happens when you have a lot of disruption within the genomic DNA, and you have deregulation of the biological processes within the cell. And these are the hallmarks of cancer that has been outlined in this classic paper, which essentially says that you have a disruption of the different signaling pathways or different growth controlling processes, or there's growth evasion, and you have inflammation that accounts for it, and all of these can give rise to tumors. So our focus here essentially is on breast cancer. And breast cancer is a very heterogeneous disease. That means no two breast cancers are alike. And they can be categorized in three groups. One is estrogen positive, that is ER positive. It can be HER2, ERBB, or new amplified group. And then we have a triple negative breast cancer, which is what our focus will be. These are the ones that lack the estrogen receptor, progesterone receptor, and HER2. Okay, all of these are lacking. So we essentially have to understand how to treat them. So just a brief outline of how cancer develops. This is a normal duct where you have cells neatly lined out. And sometimes you have hyperplasia, that means you have more cells that come. And then you have atypia of the cells, that means cells going slightly haywire. And then you have intraductal carcinoma. And then once you have this in situ carcinoma, it can evade into outside other cells, and that gives rise to invasive ductal carcinoma. So when we're talking about breast cancer, one of the genes that is highly mutated in breast cancer is APC, which was first identified in colon cancer. So what happens when APC is mutated or epigenetically silenced is it activates the Wnt beta-catenin pathway, and as a result you have development of tumor. Okay, this is an example from colon cancer, where you have loss of APC, then you have activation of other oncogenes or inactivation of tumor suppressor genes, which gives rise to carcinoma, and then you have invasion metastasis. Okay, now what we did was we wanted a system to understand TNBC. So because TNBCs lack receptors, and when we're trying to treat them, we need to treat them using chemotherapeutic agents like paclitaxel and cisplatin. So we used a cell line, which is a breast cancer cell line, MDA-MB157. And we have two knockdowns of these. And when APC is knocked down, these behave similar to TNBCs. So we looked at the transcriptomic profile of the APC knockdown for the cells that are paclitaxel resistant and for the cells that are cisplatin resistant. And we also looked at the treatments with paclitaxel and cisplatin. So we did their transcriptomic analysis using RNA-seq, and we identified several pathways that were identified using wiki pathways, and of course functional bioinformatics functional analysis. So we looked at wiki pathways, we looked at gene ontologies, and we also mapped the pathways that were relevant onto the expression levels onto the pathways. In this case, this is the Notch pathway. So the pathways that we were focused on essentially were Notch, EMT, cell cycle and cell division, IL-18, and DNA damage pathways. Okay, so what we did was we essentially looked for the pathways that were deregulated when we looked at cisplatin resistant and paclitaxel resistant cell lines in which APC was knocked down, and we looked at what happens, which are the pathways that were resistant. And we also looked at the gene ontologies for in all of these cases. So we looked at multiple ways in which we could identify, we could do the functional analysis. And then we looked at subset of genes that were at the intersection. That means they were common to both. In this case, when we compared the wild type with the knockdowns, which were resistant to both of these, and we looked at the intersection and which were unique to them. We also looked at the cell lines themselves, the wild type cell lines themselves, which are paclitaxel resistant. And we looked at different pathways, for instance, EMT, Notch, and the other pathways that we mentioned. We looked at Notch as well. And we also looked at cell cycle, which is very important, which turns out a lot of genes are disrupted. So what essentially what we have identified in this transcriptomic analysis is that we have a whole bunch of pathways that are deregulated, or genes associated with the cell cycle, and that is EMT, Notch, IL-18, and DNA damage that are regulated. So what it looks like that the APC significantly influences these pathways, and we could use these as computationally identified derived hypotheses, which we are now working on testing them experimentally. So this is our work. There's a lot of data that I haven't presented because it's huge, but for now we are using this data to do further experimental analysis.